In the last lecture, we looked at the secondary structure of proteins. If we further fold the secondary structure, we get something called as tertiary structure. Now, this tertiary structure refers to three-dimensional arrangement of amino acids on a polypeptide chain, which is responsible for conferring unique functional and structural properties to proteins. That's why you see that vegan products are much different in terms of structure and texture from their animal based counterparts. Example, the structure and texture of plant based cheese is not as appealing as their animal based counterpart. This debate has been going in food industry for a long time. They want to know how we can produce a vegan product that has a structure and texture similar to the animal based counterpart. Now, why is protein folding important? Protein folding to form secondary structure is important from a thermodynamic standpoint, as the molecule or the system always wants to be in state of lowest possible energy. So what happens is that when you fold the protein structure, there is optimization of conformational entropy of a polypeptide chain and also various interactions that are responsible for forming the structure, like hydrophobic, electrostatic, van der Waals, hydrogen bonding, and so on. The result of all this optimization is reduction in the net free energy of the molecule or system. Now, how does this reduction occur? What groups are redistributed in order to facilitate reduction in free energy? We know that protein is made out of hydrophobic residues and hydrophilic residues. The hydrophilic residues present within protein, they want to be exposed to water so that there is water hydrophilic residue interaction as they have affinity for water. Such molecules, they tend to move towards the surface of the protein. Hydrophobic residues, which want to stay away from water, they tend to get buried deep inside the protein structure. This relocation of hydrophobic and hydrophilic groups, it results in reduction of free energy. Now, numerous physicochemical properties of proteins, they are function of the proportion and distribution of hydrophilic and hydrophobic residues within a polypeptide chain. For example, we saw that proline is a alpha helix structure breaker. So if there is high amount of proline in your protein, the protein assumes a random coil structure. Generally, if your proteins, they have higher amount of hydrophilic groups, they tend to assume an elongated rod-like structure. Why? Because more hydrophilic groups can be exposed to the surface. On the other hand, if a protein has higher amount of hydrophobic groups, it tends to assume a globular spherical shape as what is depicted here. Why? Because hydrophobic group, they want to stay away from water. One important thing to remember in this regard is that around 40 to 50% hydrophobic groups, they are still present at protein water interface due to steric constraints, meaning all groups are not completely buried in the tertiary structure. So what does it imply? It implies that the whole polypeptide chain does not undergo transition to tertiary structure at a go. There are zones present within a polypeptide chain which gets folded in to form tertiary structure. The zones that form tertiary structure are called as domains. The number of domains formed is function of molecular weight of the protein. Thank you for staying with me through the video. In case you have any queries, please feel free to put them in the comment section and I'll get back at the earliest.